facilitate a panel on current research than our moderator, Amy Moore, who is the Vice President of Global Engagements and Patient Partnerships at Longevity. Amy has been involved in lung cancer space for a long, long time, has been involved with the NTRKers group from the very beginning, and was really instrumental in providing guidance and ideas and suggestions for getting the group off the ground. So Amy, so pleased to have you here as the moderator, and I'll turn it over to you.
Oh, thank you, Tim. It's been a great event so far, and I've just appreciate, Monique, your story and that of your husband and all the patients who shared their experiences this morning, and our experts who told us about the work that's going on. And I'm thrilled to facilitate a discussion with, again, some world-renowned experts in the NTRK space as we discuss the current state of where we are. And then following that, Tim will facilitate a session on future topics in the NTRK research space. So with that, I would like to introduce, and I'm sure we probably have a slide with their names and titles, but our panelists, including Dr. David Hong from MD Anderson, Dr. Herbert Leung from the Chinese University of Hong Kong, and Dr. Fernando Lopez Rios, who's with the Department of Pathology at the University Hospital in Madrid in Spain. And, I would like to ask each of you to just briefly tell us a bit more about your work in the NTRK space, what connects you to this community, and give us an orientation to, anything that you think is relevant for our audience to understand. So just brief introductions, perhaps. I see Dr. Lopez Rios first on my screen. So if you could just give us a little bit more introduction about your role and some of the work you've done in the NTRK space before we get our discussion going. Dr. Lopez Rios.
Hello, everyone. Thank you, Amy, for the introduction. Really happy to be here today with all of you, and to see some friends also in the panel. I am a pathologist, so I was involved in some of the testing in the early phase clinical trials. So that's how we started implementing the tools that we need in clinical practice to identify NTRK fusions. And then we've done, I would say, a lot of work along the lines of how can we use immunohistochemistry or next generation sequencing in a more effective manner, and come up with algorithms to make sure that we don't miss any of these patients. And I've also been very involved with ESMO. I was part of the ESMO NTRK guidelines. So I'm particularly working in lung cancer, which it's interesting because it's very challenging, as you know, to identify NTRK fusions in lung cancer. But I think it's one of the areas that I would like to talk about with Herbert and the rest of the panel.
Absolutely, and we'll come to that in just a moment. Dr. Leung, if you can introduce yourself and tell us about your work in this space.
Sure, thanks, Amy, and hello, everyone. Good morning, wherever you are. It's a little past midnight where I am, but it's great to see everyone. So I'm Dr. Herbert Leung. I'm a associate professor at the Chinese University of Hong Kong, medical oncologist by training, and my interests are actually in early phase clinical trials. And that's how I got involved with NTRK inhibitors and NTRK fusion as an entity. I'm also particularly interested in treating lung cancer as well as sarcomas. So that's where it all came in. So we were one of the initial sites to run the entrectinib trials. At that time, entrectinib was still under the company Ignita before it was actually purchased by Roche. And we were one of the institutions in the Asia Pacific region, which with Ignita's support was able to run a in-house NGS platform. And we did a landscape of about 300 patients of various cancers, and we were able to identify 3 patients to put onto the clinical trial. That's how we first started. At the same time, my involvement also included the use of larotrectinib. We're also a study site for repotrectinib. We're also a study site for PBI 200, as well as other TRK inhibitors. Of course, we all know that TRK fusions are relatively rare, but certainly for the patients who have these alterations identified, these are very good options for the patients. And more recently, actually last year, virtually, Fernando and I was part of a faculty which actually taught a advanced TRK fusion course through ESMO, and we were able to update the colleagues in the Asia Pacific region as well as in Europe on some of the latest advancements in the NTRK space. And I only just saw Fernando not too long ago in Madrid on an advisory board on how to move TRK inhibitors forward. And we'll be happy to share some of our views as well as some of our experiences with the NTRKers community.
Thank you, Dr. Leung. And I believe Dr. Hong is having some trouble connecting, but once he joins, we'll allow him to introduce himself. But until then, we'll go ahead and get started on the conversation. And so, Dr. Lopez Rios, you brought up a very important topic, and that is, how do we identify these alterations to begin with? And you've obviously written on the topic. I was part of a plain language summary that discussed the different approaches, next generation sequencing, immunohistochemistry, FISH, and I want to obviously, working in the lung cancer space at Longevity, we advocate next generation sequencing as kind of the gold standard for looking for actionable biomarkers, but we recognize that globally, maybe not everybody has the capability to run next generation sequencing. And sometimes, depending on the tumor type, maybe you can go with a different approach first. So I want you to talk us through kind of the current state of how we identify these alterations, and maybe, what some of the promise is for improving our ability to detect these alterations.
Well, yes, Amy, I think that was an excellent summary of how the field is thinking about this. I think we have to keep in mind two things that are not really opposing arguments, but okay. So the first idea is that as much as we all love next generation sequencing, and I'm completely convinced that all of our patients with not only advanced but also early stage cancer should undergo comprehensive genome profiling. That is not the reality in some institutions, many institutions in some countries, some regions, for a number of reasons. So we need to come up with more effective ways of identifying actionable targets in our patients. So in the case of NTRK fusions, we have immunohistochemistry, which I think has a very important screening role. It's not perfect, but it can help identify patients that should undergo next generation sequencing. So immunohistochemistry is still very useful. And in certain tumors, thyroid tumors, colorectal carcinomas, even lung cancer sometimes, we use it every day to make sure that we don't miss any of these patients. What I'm trying to say is that even if you run a large NGS panel, it can happen that sometimes the partner you're looking for is not included in the panel. So using immunohistochemistry as an orthogonal method, as a complementary method, is also helpful. Okay, so that's, I think, one concept. And then the second idea that I would like to discuss is the concept of integrating all the information that we have from a given patient. And that's what we do in clinical tumor boards and molecular tumor boards. So I think for me, the search for NTRK fusions is the poster child of that vision, of that change of paradigm. I think we need to work not only harder but smarter, and that happens. I think the magic happens in molecular tumor boards, because that's a place where you gather all the information that you have. You have very smart people usually in the room, from techs to all the way to data scientists or bioinformaticians, pathologists, molecular pathologists, of course, everyone involved in the clinical care of the patient. And that's where you actually realize, okay, maybe I'm in front of a patient that doesn't have an actionable target. Can I do something else? How can I go that extra mile to make sure that my patient doesn't have an actionable finding? So that's how I want to. Maybe later I can share with you a few stories. That's how we actually are now finding some of these NTRK fusions. So in summary, screening with immunohistochemistry, and then confirming all positive findings with next generation sequencing. That's strategy number one. Strategy number two, of course, next generation sequencing for everyone, which I think is not realistic as much as, again, as we are all completely convinced of this approach. And then the third idea is trying to integrate all the information, because that's how you realize that maybe you are in front of a patient with an NTRK fusion.
Right. And I see that Dr. Hong has now been able to join, so I would ask him to just briefly introduce yourself, Dr. Hong, and give us just a brief high-level summary of your connection to the NTRK space for our audience. Let's see. Dr. Hong. He may be having audio issues. So assuming that he's having some audio issues, we'll move on to the next question. And we heard from our patient representatives and participants this morning about some of the challenges. And one of the concerns that's always top of mind for any patient who's on, you know, a targeted drug or some of the newer generation drugs is what happens when that drug stops working, you know, effectively against my tumor, and kind of what comes next. So Dr. Leung, I want to talk about the topic of resistance in the NTRK space and, you know, kind of the current lay of the land when it comes to addressing resistance, and maybe also, given that you do a lot of work in the phase 1 clinical trial space, what you see currently, you know, kind of at play in combating resistance, and maybe what's in the pipeline coming next to address this challenge for our patient community.
Sure, Amy. You know, I think in terms of resistance, it's generally divided into two groups or two types of resistance. One would be a resistance along the TRK pathway, and one would be through a bypass, a resistant pathway where the tumor is no longer determined on TRK itself as one of the mechanisms of progression. And certainly in terms of the clinical trials that have been evolving ever since the development of the first generation TRK inhibitors, which are entrectinib as well as larotrectinib, we've been trying to address the issue of how to harness the issues of TRK pathway resistance. Certainly there have been drugs which have been tested. There are still some trials which are ongoing where I think the accrual has finished, but the final data is not out. But yet at the same time, there are also drugs which were in development, but then I think in terms of the readout of the results, or for one reason or another, have actually suspended development, which is a bit of a shame, really, in terms of what's being available for our patients. At the same time, though, as we're doing more next generation sequencing for our patients, we're also seeing off-target or bypass mechanisms, as I mentioned previously. So I think we're noticing that there are some patients when they have resistance to the TRK inhibitors, maybe due to other molecular alterations which have developed after the exposure to TRK inhibitors, such as there have been quoted examples of KRAS mutations, BRAF mutations, of which there are potentially other targeted therapies available. And I think there are specific cases where these patients are treated in combination with some of the other available inhibitors. And certainly TRK fusion patients are relatively rare to come by. So a lot of these cases are reported in small case reports or potentially case series. And I think we're actively trying to gather all this data as well. Apologies for my voice. Also, I guess at the end of the day, regardless of whether or not there's the presence of TRK fusion, there are also situations where the other treatments which are not related to TRK may be useful for these patients. Certainly, for example, in lung cancer, we're identifying NTRK early, so we may be using it in the early line setting. But if, for example, there is resistance and we don't have an appropriate NTRK targeted treatment, the standard chemotherapy or immunotherapy may be something that's still worth considering. So I think it's important for us to consider those options as well.
Yes, very, very much so. And I think we're seeing this kind of increasing use of combination therapies, and we'll come back to, ESMO highlighted the importance of testing for all patients, regardless of their, at least in lung cancer, regardless of their stage of disease. And we'll talk about the movement of these targeted therapies into the earlier stage setting. But I think Dr. Hong may finally have been able to connect with us. Let's see if his audio is up. Dr. Hong, can you hear us?
I can hear you guys. I was able to hear you. For some reason, there's a pretty intense firewall here at MD Anderson. Can you guys hear me?
Yes, we can. And so I would ask you just to briefly introduce yourself and, kind of the space that you operate in at MD Anderson and your connection to the NTRK space for our audience, please.
Yeah. So I'm Dave. I'm Dr. Hong, David Hong here. I'm the Deputy Chair of the Department of Investigational Cancer Therapeutics here at MD Anderson. And I was actually involved both in the phase 1 and phase 2 of larotrectinib. In fact, Sandra Montez, I think hopefully is on, was my nurse, and we were one of the first to put these patients on the studies. And in fact, I was just mentioning this to some colleagues and on Twitter that we still have a patient who's almost 10 years out on the phase 1 trial here at MD Anderson. So I've been involved in development of larotrectinib. We have tried to move forward with the development of other second generation TRK inhibitors, which was Loxo's 195, you may have recall. And also there was another molecule from a company called Pyramid Biotech, which also we were running, but unfortunately that closed. So did Loxo 195. Loxo 195 primarily because of its toxicity profile. Pyramid primarily because I think the venture capital funding issues may have affected it, and trying to determine the right dose was just going to take too long. So I agree with Dr. Long here. I think one of the challenges moving forward is, the development of kind of second and third generation NTRK inhibitors, right? And right now, many of you probably are aware, in the world of biotech, funding for new companies and new drugs is at a premium. And so unless it's a very large market like obesity, like GLP-1 agonists, et cetera, antagonists, there's not a lot of investment in this space currently. Hopefully this will change in the future, but right now there's not been a whole lot of investment into the space.
Right. And we heard a bit about that earlier in the first half of the summit this morning, and just the challenge of developing new drugs in the rare cancer space. And so, that's one of those bigger challenges that I think across the cancer community we're grappling with, and how to address it to make sure that all patients can benefit from the scientific advances that we're seeing in the cancer space and in the NTRK space. So thank you for that. So I want to, you brought up the fact that you have a patient who's 10 years out on the phase 1 clinical trial, and Sandra was on earlier. She facilitated a couple of our sessions from Australia. So she's a trooper for doing that when it's very early her time. But the point being, we're now seeing long-term survivors who are, living for years on TRK inhibitors. And so, some of the conversation was around some of the side effects that patients are managing through as they're living longer, and the balance of tolerability for when some side effects, when you were only looking at maybe a 1 year survival, you could kind of wrap your mind around that. But now when you're facing luckily surviving for 6, 7, 10 years, it changes the equation perhaps. And so what are some of the advances that we're seeing currently in managing these side effects that occur on TRK inhibitors? So Dr. Lopez Rios and Dr. Long, you taught this course and have been intimately involved with the guidelines around management of NTRK positive disease. Do you have thoughts on managing the side effect profile for patients?
Yeah, I think one of the biggest improvements, I think, is the recognition of these side effects. I think as we have more of these drugs available, more patients being exposed, initially, again, TRK positive patients are few to come by, but as there's more experience, it's being available in the market commercially, we do recognize that there are some distinct side effects, which I think initially it's really the first exposure for oncologists to experience. Weight gain being a side effect. It's something that initially, first, a lot of these patients, when they were put on the study, they were actually already quite ill, they were quite sick, and then there was weight gain, everybody was very happy. But then suddenly you realize they're actually gaining much more than what you would expect normal. And certainly that's something that we had to deal with. Some of the neurocognitive side effects, that initially exhibits itself probably in the first 1 or 2 months. That's something that needs to be recognized by both the patients as well as the caregivers. But thankfully, I think for that, as time progresses, as patients are exposed to the drugs for a longer period of time, they do tend to settle. So I think with that, it also therefore gives confidence to the physicians as well as for the patients and the caregivers that there are these side effects which can be present, but after some dose interruption, some adjustments, and some longer exposure, they tend to go away. So I think that's something that was only possible through gaining experience.
Did you want to add anything, Dr. Lopez Rios?
I'm afraid not on this topic because I don't see I don't treat patients. But I like maybe to briefly answer one of the questions that we had, if you think that's appropriate, Amy.
Yes, please go ahead.
Yeah, I think in terms of understanding the mechanisms of resistance, which are now new therapeutic opportunities for our patients, I think very important to emphasize repeat testing using large and robust NGS panels. I personally think we have to try to use both tissue and plasma and blood, but whatever you do, it's fine as long as you do it. I think because we're still struggling trying to understand what those mechanisms are. So you need as much information as possible. So that I think that's a very important message, and sometimes that message needs to be repeated at clinical tumor boards and molecular tumor boards. So it's not okay to have just one single NGS result. You need sometimes to follow up and really understand the molecular context of the tumor.
Absolutely. I have a couple more questions, and I know we only have a few more minutes for our panel, but one is for you, Dr. Lopez Rios, on this topic of, we're seeing this migration of targeted therapies and immunotherapy into the early stage space. And so that puts, the impetus on all of us to, test our patients when they have this diagnosis, even for patients with early stage disease. And so how can we work with pathologists to ensure that this testing is happening, not just in the advanced stage setting, but even for patients with early stage disease?
I think it's very important that we see the amazing clinical results that are happening now in the space. This is a huge change in the way we approach not only target therapies but also immune checkpoint inhibition. And I always tend to say that now early stage patients are helping advanced stage patients, because if you think of it, the way we work in the lab, we need to to optimize our lab workflows, our NGS chips. You know, we run next generation sequencing. It's a pity I don't have one of those small chips. It's a 2 square centimeter plastic thing, and we need to optimize those workflows. Otherwise, it's impossible to maintain the cost even in a national health system of these comprehensive genomic efforts. So I think if we start considering early stage patients as well as as advanced stage patients, we will be able to help more patients. And this is a concept that unfortunately is not, I think, well advertised and recognized outside molecular labs. So I would do those two things. I would make sure lab people, you know, like I'm doing today with these with NTRKers, and I love that motto, no, testing, knowledge, and support. I think knowledge actually triggers testing, so we need to know about these amazing clinical results, and then the other way around. I think it's a two-way education effort. I think people that make decisions, they need to understand that if we include early stage patients, that's going to benefit everyone. So so I think it's happening, and I'm very optimistic.
Great.
Amy,
Dr. Hong, I'm sorry. Go.
Yeah, I wanted to just comment on I don't know if I could comment briefly on the side effect issue.
Yes.
And then come back to the testing. So you know, we don't fully understand I mean, there's three main toxicities that we see. We see these neurotox, the dizziness, the kind of the pain reflex. Then we see the weight gain, and then we see theoretically the bone fractures, et cetera, right? And I would say that the bone fracture issue to date has really been reported more so with entrectinib rather than larotrectinib. And I don't know, you know, that may be something that you may want to consider. If you're on entrectinib, you're responding, you may want to consider, and you're having bone fracture issues, you may want to consider switching over to larotrectinib. The other thing was the other two issues, the weight gain. We don't fully understand that. We know that inhibition of TRK modulates, you know, these things, and we're not sure whether that's truly because of, you know, people, their appetites increase, or just overall other ways in which people gain their weight. And you know, right now the problem is one of the challenges is that the few number of patients, and can we use something like GLP agonists in this case to obviate the weight gain and so forth? That I think it would be worth trying, but it's a difficult study to do in this patient population. The last issue was the neuropathy, and one of the reasons that these drugs like LOXA 195 has failed is because of the neurotoxicity, the dizziness, the whatever, and we still don't understand that. We think it has to do with TRK-B, which binds to something called BDNF, and we think that it may have to do with either an increase in BDNF or a downregulation of BDNF. And BDNF is a ligand for GABA receptors. And I explored this here with Dr. Maj, who's a neuro-oncologist here. And we tried GABA agonists, such as benzodiazepines, et cetera, and it didn't really seem to affect the overall neurotoxicity. So we still have a lot to learn, but I think that we could probably figure this out. It's just a matter of just understanding a little bit more in depth about this, the BDNF pathway. So that's I just wanted to clarify that, because there is some research already going on in this space.
And thank you for all those comments. Very thoughtful. And I do want to be respectful of our timeline here, and I know we need to wrap this up. But again, I want to extend my gratitude to all three of you. You're leading experts in this space, and it's such an honor to have you with us, sharing your expertise for our community. And I know we look forward to seeing continued progress in this space, and I'm very hopeful with people like you leading the charge that we'll continue to see progress and improve outcomes for our patients with TRK fusion cancer. So thank you again for being here.
Thank you very
Yes,
much.
Thank you.
thanks to all of you. And I know some of you are able to join us for the later Q&A session that'll be in about half an hour after we do this next panel. Some of you may not be able to understand time zones and other obligations and things, but if you can join us, that would be lovely. Amy, thank you so much for facilitating this discussion, and really good to hear that the things that I think we heard this morning being bubbled up from people saying, I'm concerned about this, I'm concerned about this, I'm concerned about that. We're beginning to get insights into, we're beginning to get some answers. And to me, to have Dr. Hong talk about having somebody who's been on this treatment for 10 years is quite amazing. So thank you very much.